start, and also remember that every year NTA is asking either one, or if you are lucky you can even get 2 questions from this chapter. Cellular respiration, how respiration process is executed within a cell. Cell, that means protoplasmic respiration is taking place when the cell is actually starving. But if there is no oxygen into the cell, then what will happen? So this is substrate level phosphorylation, SLP. So CO2, water and a high amount of energy will be released. So succinic acid will convert into malic acid, which is a 4-carbon compound. So no carbon will be coming out in form of CO2. So listen this carefully. Whenever your respiratory substrate is a carbohydrate, let's say glucose, the RQ is going to be 1. So you have to first understand your numbers kaise aaye, and then you have to remember also for easily and fastly giving the answers. Hello everyone, my name is Kiran Chaudhary and I welcome you back at Infinity Learn by Sri Chaitanya. My dear students, in this class we will be discussing about the complete chapter respiration in plants. So I'll teach you all the important concepts and facts on which NTA has already asked question and will be asking. There's a high chance that the information, the facts, the concepts that I will be making you revise in this lecture, there's a high probability that you might get a question in your coming NEET 2023 as well based on today's lecture. So let's start, and also remember that every year NTA is asking either one, or if you are lucky you can even get 2 questions from this chapter. So let's get started. Okay. So as you can see, first thing I would like to explain you is what respiration means and what exactly this chapter deals, or this chapter is all about. So this chapter is about respiration, and to be more specific my lovely kids, इस chapter में हम लोग बात करेंगे cellular respiration के बारे में. Cellular respiration क्या होता है? Cellular respiration एक रेस्पिरेशन प्रोसेस है जो कि सेल के अंदर चल रहा है. तो हम लोग यहाँ पे टिश्यू, ऑर्गन इस लेवल पे बात नहीं करेंगे, राइट. मोरोवर प्लांट्स के अंदर होते भी नहीं हैं, राइट. सो वी टॉकिंग अबाउट रेस्पिरेशन इन प्लांट्स, एंड इफ वी टॉक अबाउट प्लांट्स, हियर आई विल बी टीचिंग यू cellular respiration, how respiration process is executed within a cell. So remember, basically in this chapter we will try to understand that how a particular respiratory substrate, which can be a carbohydrate, example glucose, or fatty acid, fats or proteins, etc. So how these respiratory substrate will be broken down. Remember, respiratory substrates are made up of carbon, right? There are several carbon-carbon bonds in them. So during cellular respiration, my dear students, inside a plant cell, carbon-carbon bonds between a respiratory substrate will be broken down. As a result, there will be release of energy, and this energy will be released in slow steps. The steps will be slow enough that the released energy can be trapped into ATP molecules. So this is basically cellular respiration, where I will teach you how a particular respiratory substrate will break and the release of energy, how it will lead to formation of ATP molecules which are required to do several vital functions inside a cell, which is required to maintain the living state of a particular cell or the organism. So let's get started, my lovely kids. So all what I have explained right now has been explained in 2 lines of the definition of cellular respiration. I can also read out for you. So cellular respiration is a mechanism of breakdown of food material within the cell to release energy and trapping this energy for synthesis of ATP. So I have elaborately explained this line. Now next thing is respiratory substrate. So what type of substances can be oxidized during cellular respiration process in order to release energy which can be trapped into ATP molecules? So the respiratory substrate can be carbohydrates, fats and proteins, and even under special conditions inside plant cells, organic acids can also act as respiratory substrate. Now, if we talk about the most common respiratory substrate, please remember it is the glucose. So, most common is the glucose, and that is why if you remember and if you try to recall The entire process of cellular respiration in your NCERT is also explained using glucose as the respiratory substrate. So let's try to understand that, but there are few other points as well to understand. The first thing is, do plants breathe? So listen, my dear students, definitely each and every cell of the plant which is living will show respiration, and during respiration plants will consume oxygen and release CO2, right? But plants do not require respiratory organ. So there is no respiratory organ or respiratory system inside a plant cell. Why? Because if I talk about the requirement of gaseous exchange is quite less, and the distance the gases has to travel is also less, and moreover each and every cell or tissue of the plant body can take care of its gaseous needs. So that is why plants can get along without respiratory substrate. But remember, there is utilization of oxygen and release of CO2 during cellular respiration process in each and every cell of the plant, whether you talk about leaf or you talk about root. Next thing, my lovely kids, so whatever I am explaining, it's like that I explain a point and the same point according to NCERT line will be displayed on the screen. So you can pause the video and read out. So I'm also trying to come out of the frame so that you can pause, screenshot or read what is written on the slide. Same thing I have explained in elaborated form. Next is, my dear students, types of respiration. So remember kids, based on respiratory substrate. So what is the, or in which form the respiratory substrate is present in a plant cell, the respiration can be of two types. First is the floating respiration. What is a floating respiration? Whenever the respiratory substrate is present in a stored form or in form of stored material inside the plant cell, then that type of respiration is called as floating respiration, and here the respiratory substrate can be carbohydrates, proteins or fats, any, but remember they all will be in form of stored material. Another type is the protoplasmic respiration, and in protoplasmic respiration, my lovely kids, what will happen? The structural and enzymatic proteins, the proteins which are part of a, let's say, cell membrane or an enzyme, these types of proteins will be utilized during cellular respiration process. These types of structural and enzymatic proteins will be broken down during, my dear students, protoplasmic respiration. But remember, protoplasmic respiration takes place when there is absence of stored food or stored material inside the cell. That means protoplasmic respiration is taking place when the cell is actually starving. So these are two types of respiration, floating and protoplasmic, based on the respiratory substrates. So you can pause and read out. Next thing we need to understand, my lovely kids, is types of anaerobes. So there are basically two types of anaerobes. One is, my dear students, facultative anaerobe, and another is obligate anaerobe. Now remember the term facultative. Facultative means it is optional. Obligate means it is like it's a foundation, right? So facultative means it's optional, and obligate means there will be some compulsion. So facultative anaerobes will be, my dear students, optional anaerobes. That means facultative anaerobes will be actually aerobic organisms which can respire even in the absence of oxygen. But when we talk about these obligate anaerobes, they are compulsorily anaerobic organisms. These obligate anaerobes will respire only in the absence of oxygen, or rather I should say obligate anaerobe will respire without using or consuming oxygen. In fact, these obligate anaerobes will lack the enzymes necessary for conducting aerobic respiration. So facultative anaerobes are aerobic organism which can respire even in the absence of oxygen. Obligate anaerobe will respire without using oxygen compulsorily. Next. Okay, so now let's start with the main thing that's why we are conducting this session, that is the cellular respiration process. So remember, in this entire chapter given in your NCERT is trying to explain you cellular respiration by considering glucose as the respiratory substrate. So right now I will start explaining you glycolysis, then I will explain you pyruvate oxidation, then I will or link reaction, then I will explain you TCA cycle, right, or Krebs cycle. So basically we are considering glucose as the respiratory substrate and then explaining the entire cellular respiration process. So remember, my dear students, what will happen? First of all, glucose will enter inside the cytoplasm, right? So, or I can say that the stored form of the food which is present inside a plant cell, those two stored form of the carbohydrate will convert with the help of certain types of specific enzymes or certain types of reactions. These stored carbohydrates will convert into glucose. So from here we will start our discussion. So first of all, glucose will undergo glycolysis within the cytoplasm. So during glycolysis, what will happen? Glucose will break down into two molecules of pyruvic acid. Glucose is a 6-carbon compound, pyruvic acid is a 3-carbon compound. So from one molecule of glucose, my dear students, during glycolysis, which include 10 different reactions, my dear students. So because of 10 different reactions the glucose is undergoing, finally it will get converted into two molecules having 3 carbon each, pyruvic acid. Now this is glycolysis, which is a common pathway whether organism is doing aerobic or anaerobic respiration. Why? Because first of all, this glycolysis do not require oxygen. So for glycolysis to take place, there is no requirement for oxygen to be present in the cell or around the cell. So that means glycolysis can take place in the presence as well as in the absence of oxygen. So glycolysis is a common pathway between aerobic and anaerobic organism. This should be something very much clear in your head. Now the second thing that should be, my lovely kids, clear in your head is, now what will happen to the pyruvic acid? What will be the fate of pyruvic acid? So just don't try to get lost, just try to listen me carefully. If you get distracted, just close your eyes and try to listen as much as possible. So I'm just trying to explain you everything in an overview format in a story. Okay, so listen that first of all, glucose, your respiratory substrate, has undergone glycolysis and converted into two molecules of pyruvic acid. This is a common pathway, can take place during anaerobic as well as aerobic respiration. Right now, if there is presence of oxygen in the cell, then the pyruvic acid will enter into the mitochondrial matrix and continue with the aerobic respiration. But if there is no oxygen into the cell, then what will happen? The pyruvic acid will undergo anaerobic respiration. So during anaerobic respiration, two types of fermentation can take place as given in your NCERT, lactic acid or alcoholic fermentation. Now listen, let's try to understand, let's try to imagine if there is no oxygen in or around the plant cell, then what will happen? The glucose which has undergone glycolysis and produced two pyruvic acid, now each pyruvic acid, if it enters into lactic acid fermentation, every pyruvic acid will convert into one lactic acid by consuming one NADH plus H plus molecule. So there will be production of two molecules of lactic acid from one molecule of glucose by consuming two molecules of NADH plus H plus. Now if the plant cell is using the alcoholic fermentation, what will happen? One molecule of glucose, my dear students, after undergoing glycolysis will produce 2 pyruvic acid, and these 2 pyruvic acid, my dear students, every pyruvic acid during alcoholic fermentation will convert into 1 molecule of ethanol or alcohol and will produce 1 CO2 also, because pyruvic acid is a 3 carbon compound, ethanol is a 2 carbon compound. So 1 carbon from each pyruvic acid comes out in form of CO2. So 2 molecules of pyruvic acid produced at the end of glycolysis from 1 molecule of glucose will undergo alcoholic fermentation to finally produce 2 molecules of ethanol, 2 molecules of CO2, and will consume 2 molecules of NADH plus in every conversion of pyruvic into the alcohol, right? So this is all about anaerobic respiration, lactic acid or alcoholic fermentation. Now let us try to recall, let's say if there is oxygen present into the cell, what will happen? So in the presence of oxygen, the plant cell will use or will undergo aerobic respiration. In that, my dear students, 1 molecule of glucose which has undergone glycolysis and produced 2 molecules of pyruvic acid, both the pyruvic acid molecules will enter into the mitochondrial matrix in the presence of oxygen, and as a result what will happen? The pyruvic acid will first undergo link reaction where pyruvate will be oxidized, a 3 carbon molecule will convert into a 2 carbon molecule acetyl-CoA, and there will be release of 1 CO2 from each pyruvic acid molecule in every step of in every single link reaction. Now this acetyl-CoA will react with oxaloacetic acid and start the Krebs cycle where 2 molecules of CO2 will be released, 3 molecules of NADH plus H plus, 1 molecule of FADH2, and 1 ATP or GTP will be produced in every turn of Krebs cycle. Similarly, this turn will take place twice because there is formation 2 acetyl-CoA because there were 2 pyruvic acid produced at the end of glycolysis. So this is the overview of aerobic and anaerobic respiration. I wanted you to listen this first so that whenever I am going to actually explain it with the help of PPT, you will be revising it twice, and maybe this will help you to retain this information for a longer period of time. So let's get started now. Again, I will make you revise, don't worry. So glycolysis, it will be beneficial if you try to rewind this, go back and try to listen all this explanation again by closing your eyes. This will help you to imagine better, retain better. Glycolysis. So glycolysis kahan hota hai? Glycolysis will take place in all living organisms. Why? Because देखो सिंपल सी बात है ना, ग्लाइकोलिसिस कहाँ हो रहा है? साइटोप्लाज्म में, और साइटोप्लाज्म तो सभी लिविंग ऑर्गेनिज्म के पास में है, whether it is a prokaryotic cell or a eukaryotic cell. Next, oxygen is not required for glycolysis to take place, so it's a common pathway between aerobic and anaerobic organism. So whether organism is showing fermentation or aerobic respiration, it will definitely show glycolysis, whether you talk about aerobic or anaerobic respiration. During glycolysis, glucose will be broken down partially into 2 molecules of pyruvic acid. So glucose is a 6 carbon compound, pyruvic acid is a 3 carbon compound. So these are the 10 reactions of glycolysis. Let's get started. So in the first step, what will happen? First step is that, or rather I can say this is the first phase of the cellular respiration. This is the first phase of glycolysis, my lovely kids, where activation of the glucose molecule will be taking place, right, or energy will be invested. So there will be investment of energy, there will be activation of the glucose molecule in the first phase of the glycolysis. This is the first phase of glycolysis. So in the first, we will start with glucose. So glucose is a 6 carbon compound. It is converting into glucose 6-phosphate. So there is a phosphorylation. There is addition of a phosphate group into a glucose molecule, and this phosphate group will be added by using or by doing the expenditure of 1 ATP molecule. So ATP molecules will convert into ADP, and inorganic phosphate will be added into the glucose molecule. So glucose will be activated. Now this glucose phosphate will convert into fructose 6-phosphate. Acha, the enzyme in the first step is important. This is hexokinase activated by Mg2+ in the first step. So this is your first step. In the second step, isomerization will take place. Glucose 6-phosphate will convert into fructose 6-phosphate. Now, in the third step, which is a rate determining step, my dear students, or rate limiting step, here fructose 6-phosphate will be phosphorylated to form fructose 1,6 biphosphate. So that means there will be 2 molecules of phosphate attached in this molecule of the fructose 1,6 biphosphate. This activity or this reaction will be done in the presence of phosphofructokinase, also known as PFK, and the enzyme will be activated by Mg2+. So here also the additional phosphate group is added to fructose 6-phosphate to produce fructose 1,6 biphosphate by doing the expenditure of 1 ATP. This is the third step. So what you should remember, that in glycolysis, in first and the third step, activation will be done, energy will be invested. So there will be expenditure of 1 ATP each in step number 1 and step number 3. Remember, step number 1 and step number 3 are also irreversible. Next, fructose 1,6 biphosphate were split into 2 molecules. Still here, all the molecules were of 6 carbon. Now this fructose 1,6 bisphosphate or biphosphate, which is of 6 carbon, will split into 3 carbon compound, 2 3-carbon compound. So this will be your fourth step, right? So where glycerol, glyceraldehyde, slip of tongue, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate will be produced. So there will be formation of 2 molecules when fructose 1,6 biphosphate is breaking. So there will be production of glyceraldehyde 3-phosphate as well as there will be production of dihydroxyacetone phosphate. Now in the fifth step, dihydroxyacetone, this DHAP will back convert into glyceraldehyde 3-phosphate. So from here, every reaction from here, so this is the fifth, now this is the sixth step. So from the sixth step, every reaction will be multiplied by 2. Every reaction will take place twice. Every product, every reactant will be multiplied by 2, because once the reaction 6, 7, 8, 9, 10, once the reaction will take place for glyceraldehyde 3-phosphate and another time it will take place for this DHAP, which has converted into glyceraldehyde 3-phosphate. So notice this. So this is the sixth reaction, right? And in the sixth reaction, what will happen? This glyceraldehyde 3-phosphate, the aldehyde will convert into acid and there will be addition of a phosphate also. So here glyceraldehyde 3-phosphate is having 1 phosphate group, but here in this molecule, in this acid, phosphoglyceric acid, there are presence of 2 phosphate. That means in the sixth step, phosphorylation will take place and aldehyde will be converted into acid. I hope this is clear. So here in the sixth step, the additional phosphate which is added will not be coming from the ATP molecules. Rather, here the source of the inorganic phosphate is H3PO4. So there will be requirement of 2 H3PO4 to convert 2 molecules of phosphoglyceraldehyde phosphate into 2 molecules of 1,3 biphosphoglyceric acid. And in the sixth step, not only phosphorylation is taking place, in the sixth step there will be production of NADH plus H plus molecules also. So since the sixth step will take place twice, there will be production of 2 molecules of NADH plus H plus in the sixth step. So in first and third step, ATP is utilized, phosphorylation taking place. In the sixth step, again phosphorylation is taking place, but along with that NADH plus H plus is also getting produced. Here in the sixth step, ATP is not utilized. Now in the next, what will happen? 1,3 biphosphoglyceric acid will break down into 3 phosphoglyceric acid. इसका क्या मतलब है? Simple, यानी कि एक phosphate group, 1 phosphate group will be removed. And this phosphate group which is removed will be accepted by ADP molecule to produce ATP. So this is substrate level phosphorylation, SLP. तो क्या है मेरे प्यारे बच्चों, ये है SLP, सबस्ट्रेट लेवल फॉस्फोराइलेशन. तो क्या होता है इस सबस्ट्रेट लेवल फॉस्फोराइलेशन के अंदर? तो एक सबस्ट्रेट लेवल फॉस्फोराइलेशन के अंदर मेरे प्यारे बच्चों, देयर विल बी रिलीज ऑफ one substrate will convert into another substrate, or one substance will change into another substance, and there will be release of energy as well as there will be release of the inorganic phosphate. So the inorganic phosphate will be accepted by the ADP molecules to produce ATP. So here the ATP production is taking place without forming the proton gradient, by just converting one substrate into the another one substance into the another one. This is substrate level phosphorylation. Since this step, right, the 7th step will take place twice, so there will be production of 2 ATP in the 7th step using substrate level phosphorylation. Now in the 8th step, what will happen? 3 phosphoglyceric acid, 3 the position of the functional group. So 3 phosphoglyceric acid will convert into 2 phosphoglyceric acid rearrangement, and then my dear students, what will happen? 2 phosphoglycerate will convert into phosphoenol pyruvate. Now here comes your 10th step, which is the most important step. So in the 10th step, again there will be substrate level phosphorylation, that is SLP. Here each molecule of phosphoenol pyruvate will convert into pyruvic acid or pyruvate, and there will be release of inorganic phosphate along with some energy that will be accepted by ADP molecules to produce ATP. Since this 10th step will also take place twice, there will be production of 2 ATP molecules in the presence of pyruvate kinase. You can go through this, pause the video and notice. 6, 7, 8, 9, 10 steps will take place twice. So listen this guys, in the entire glycolysis process where 10 reactions are taking place, 1 molecule of glucose is breaking down into 2 molecules of pyruvic acid. There are total, so there will be total. Total means in the entire glycolysis I am talking about. In the entire glycolysis I am talking about. So total ATP कितना प्रोड्यूस होगा? Total ATP 4 है. So 2 ATP in the 7th step and 2 ATP in the 10th step will be produced. So total ATP बनेगा 4. But remember, -2 आपको करना पड़ेगा. Why -2? Because 2 ATPs were consumed. 2 ATPs were consumed in the first and the third step, right? और यहाँ क्या हो रहा है? 7th और 8th पे production हो रहा है, 1 और 3rd step पे consumption हो रहा है. So if somebody asks you what is the net gain of ATP at the end of glycolysis, you should say 2 ATPs are produced. This is the net gain, not total production. Total production तो आपका 4 का है. बट आप नेट कैसे निकालेंगे? प्रोड्यूस्ड माइनस कंज्यूम्ड. सो प्रोड्यूस्ड माइनस कंज्यूम्ड इज द नेट गेन विच इज of 2 ATP, and along with that there will be production of 2 NADH plus H plus molecule. तो ये NADH plus H plus molecules भी बनेंगे कहाँ पे? 6th step पर, right? So what you need to understand is at the end of glycolysis, from 1 glucose molecule you are getting 2 molecules of pyruvic acid, 2 ATP का net gain and 2 NADH plus H plus molecules. This is something you got to remember. Next, so as I have already explained, what is this fate of this pyruvic acid which is produced at the end of the glycolysis? So this pyruvic acid, depending upon the condition present in the cell, if there is presence of oxygen, aerobic respiration will take place. So this pyruvic acid will be completely broken down, all the carbons will be released, all the 6 carbon present in 1 molecule of the glucose will be released in form of 6 CO2 molecules at the end of the aerobic respiration, my dear students, that will take place inside the mitochondria, right? So in aerobic respiration, where I will explain you, my dear students, pyruvate oxidation or link reaction and citric acid cycle. So combining two, you will see there will be release of all the 6 molecules of CO2. Complete oxidation, complete breakdown of glucose will take place, complete breakdown of respiratory substrate will take place in the presence of oxygen. So CO2, water and a high amount of energy will be released. And in the absence of oxygen, what will happen? Fermentation or anaerobic respiration will take place. So in alcoholic fermentation, what will happen? Alcohol, CO2 and a very less amount of energy equivalent to 2 ATP will be released, and in lactic acid fermentation, lactic acid plus very less amount of ATP energy, that is 2 ATP will be produced, right? So this is about fate of pyruvic acid. So in the absence of oxygen during anaerobic respiration, first of all, there will be incomplete breakdown of respiratory substrate. So whenever glucose is taking the anaerobic pathway in form of lactic acid or alcoholic fermentation, glucose will not break down completely. All the bonds of the carbon-carbon present in the glucose molecule will not break. As a result, there will be a release of less energy, and even less than that will be trapped into ATP. But in aerobic respiration, there is complete breakdown of the glucose molecule or respiratory substrate. So a lot of energy is released stepwise, as well as a lot of ATP will be produced. Next is, my dear students, let us try to take a small overview about the major anaerobic pathways that I have already explained. So this is your anaerobic pathway. This is your anaerobic respiration. ठीक है, अब देखते हैं. तो सबसे पहला है, जैसे कि मैंने आपको बताया, anaerobic respiration major pathway, it will be lactic acid or alcoholic. So remember, for lactic acid or for alcoholic fermentation, glycolysis is common, right? Whether it is aerobic or anaerobic, glycolysis will take place. So here I am talking about the major pathways of anaerobic respiration. First of all, glycolysis will take place. ये पूरा आपका क्या है? Glycolysis है. ये पूरा मेरे प्यारे बच्चे आपका glycolysis है. One molecule of glucose is breaking down into 2 molecules of pyruvic acid, and there will be production of 2 molecules of NADH plus H plus, and there will be net gain of 2 ATP also in the glycolysis. Now during lactic acid fermentation, so during lactic acid fermentation, what will happen? Each pyruvic acid, which is a 3-carbon molecule or compound, will convert into lactic acid, which is also 3-carbon. Hence there will be no release of CO2 molecules during lactic acid fermentation, but there will be consumption of 1 NADH plus H plus, right? So whenever 2 molecules of pyruvic acid, which is produced at the end of glycolysis, they will enter into lactic acid fermentation, they will produce 2 molecules of lactic acid by consuming 2 molecules of NADH plus H plus. So these 2 NADH plus H plus are actually coming from where? From the glycolysis. So they are cancelled out. मतलब glycolysis में आपने 2 NADH plus H plus बनाए और फिर lactic acid fermentation में उनको consume कर लिया. So at the end of lactic acid fermentation, this anaerobic pathway, you will say there is a net gain of just 2 ATP which is produced, my dear students, in the glycolysis. यहाँ तो कोई ATP बन नहीं रहा, और इसके साथ साथ जो NADH plus H plus आपने 2 बनाए थे during glycolysis, they are also consumed. Now next is your alcoholic fermentation. So what will happen in alcoholic fermentation? Pyruvic acid will finally break down into alcohol or ethanol, which is a 2-carbon compound. So when 1 pyruvic acid finally breaks into ethanol, there will be production of 1 ethanol as well as 1 CO2 molecules. So in lactic acid, there is no production of CO2, but in alcoholic fermentation, there will be release of CO2 molecules. So 2 pyruvic acid molecules which are produced at the end of glycolysis will produce 2 ethanol along with that 2 CO2 molecules. And remember, in every alcoholic fermentation reactions, one NADH plus H plus will be consumed. So that means for the formation of 2 ethanol molecule, there will be utilization of consumption of 2 NADH plus H plus. So similarly, just like in lactic acid fermentation, my dear students, the NADH plus H plus which are produced during glycolysis, both of them will be consumed during alcoholic fermentation to produce 2 molecules of ethanol. So at the end of even this anaerobic pathway, alcoholic fermentation, there will be just net gain of 2 ATP. The NADH plus H plus that were produced in the glycolysis are also consumed. Both of them are consumed during alcoholic fermentation. So finally, what you should say, during anaerobic respiration like lactic acid fermentation or alcoholic fermentation, there is a net gain of just 2 ATP, net gain of just 2 ATP. So here I have shown you in a little elaborative form how these reactions takes place. So this is your lactic acid fermentation. Where pyruvic acid is breaking into lactic acid, and there will be also consumption of NADH plus H plus molecules, and here NAD positive will be released, right? This will take place in the presence of lactic acid dehydrogenase and Zn2 plus ions, and this is your alcoholic. This is your alcoholic fermentation. So if you see in alcoholic fermentation, what will happen? First of all, the pyruvic acid will convert into acetaldehyde. The pyruvic acid will convert into acetaldehyde, and in the presence of pyruvic acid, my dear students, pyruvic acid decarboxylase and Mg2 plus. So in the first step, your CO2 molecules will be released, and in the second step, the acetaldehyde will be converted into ethanol, and here the utilization of NADH plus H plus will take place. This reaction will be catalyzed by alcohol dehydrogenase in the presence of Zn2 plus ions. So this is all about the major pathway of anaerobic respiration. You can pause the video, take a screenshot, or just try to recall what all I have explained over here. Next is aerobic respiration. चलिए समझने की कोशिश करते हैं किस तरीके से होता है आपका एरोबिक रेस्पिरेशन. सो एरोबिक रेस्पिरेशन कब होगा? जब आपके सेल में या सेल के सराउंडिंग में ऑक्सीजन प्रेजेंट है. सो द प्रोडक्ट्स ऑफ ग्लाइकोलिसिस, दैट मीन्स द 2 molecules of pyruvic acid will enter into the mitochondrial matrix in order to undergo aerobic respiration. So majorly हम लोग 3 प्रोसेसेस के बारे में बात करेंगे, स्टेप्स के बारे में बात करेंगे, राइट. फर्स्ट इज फॉर्मेशन ऑफ एसिटाइल कोए. तो इसमें क्या होगा? सो दिस इज एक्चुअली योर लिंक रिएक्शन और पायरुवेट ऑक्सीडेशन. सो ड्यूरिंग लिंक रिएक्शन और पायरुवेट ऑक्सीडेशन which is taking place in the mitochondrial matrix, one molecule of pyruvic acid will break down into one molecule of pyruvic acid will break down into one molecule of acetyl CoA, and one CO2 molecule will be released. So at the end of at the end of link reaction, which will take place twice for 2 molecules of pyruvic acid, there will be production of 2 acetyl CoA made up of 2 carbon each. Now this acetyl CoA, which is produced at the end of the link reaction, will react with oxaloacetic acid, the first member of the citric acid cycle or TCA cycle, to start the cycle and produce the first product of TCA cycle, that is the citric acid, right? So finally at the end of TCA cycle, all the carbons present in a respiratory substrate will be released in form of CO2. So complete breakdown of the respiratory substrate will take place at the end of TCA cycle. Now the energy that is released during breaking the carbon-carbon bonds of the respiratory substrate, since it is released slowly, right? So the energy that is released in breaking these carbon-carbon bonds of the respiratory substrate, they will be trapped to form high energy chemical intermediates like ATP, right, via SLP, substrate level phosphorylation, many molecules of NADH plus H plus and FADH2. Now the next step is the terminal oxidation, which will take place with the help of electron transport system, which is present in the inner mitochondrial membrane. So what will happen there? So in the inner mitochondrial membrane there is ETS present, there are several electron carriers present in form of complexes. They will help to break NADH plus H plus and FADH2 molecules. These are high energy chemical intermediate. They carry a lot of energy, higher than that what is there in 1 ATP. So while breaking down, while oxidizing these NADH plus H plus and FADH2 molecules, again there will be release of energy and there will also be build up of the proton gradient. So because of the proton gradient being built up between the outer and inner mitochondrial membrane, whenever this proton gradient will break, it will release energy, and the energy release can be utilized to make several molecules of ATP using chemiosmosis, right? So all this I will be explaining in the coming minutes. So let's start with the first, formation of acetyl-CoA and TCA cycle. So remember, this is your formation of acetyl-CoA, or you can say link reaction. Okay, so and this is your TCA cycle or Krebs cycle, and remember all this is taking place in the mitochondrial matrix. Alright, so first of all what will happen? Pyruvate oxidation. That means pyruvate, which is made up of 3 carbon, will convert into acetyl-CoA, which is made up of 2 carbon. So there will be release of 1 carbon in form of CO2, and there will be production of 1 NADH plus H plus in every link reaction. Since there are 2 pyruvic acid produced at the end of glycolysis, so there will be 2 link reactions. There will be formation of 2 acetyl-CoA, 2 NADH plus H plus and 2 CO2, right? Next is, my dear students, citric acid cycle. So every acetyl-CoA will react with oxaloacetic acid, which is the first member of TCA cycle. Oxaloacetic acid is the first member. So oxaloacetic acid, which is a 4 carbon compound, will react with acetyl-CoA to produce, which is a 2 carbon compound, to produce a 6 carbon product known as citric acid, which is the first product of citric acid cycle, and hence the name citric acid cycle. So this citric acid cycle, which is made up of 6 carbon, will convert into alpha ketoglutaric acid, and which is a 5 carbon compound. So that means 1 carbon from citric acid will be released in form of CO2. So remember, try to understand, wherever the carbon is coming out in any step, in any reaction, wherever the carbon is coming out in form of CO2, that means you try to understand the respiratory substrate is breaking and the carbon is releasing, right? Energy is released, right? So here, my dear students, what will happen? 1 molecule of NADH plus H plus will be produced when citric acid breaks into alpha ketoglutaric acid, along with release of 1 CO2 molecule. Alpha ketoglutaric acid is a 5 carbon compound. Through several reactions, it will convert into succinic acid, which is a 4 carbon compound. So again, 1 molecule of carbon, 1 carbon will come out in form of CO2, right, and 1 NADH plus H plus molecules will be produced again, and also there will be production of GTP molecule. So there will be production of succinyl-CoA over here, right? So succinyl-CoA will be produced somewhere over here in between, and whenever a succinyl-CoA converts into a succinic acid, there will be formation of 1 GTP molecules, and this is SLP, substrate level phosphorylation, right, where succinyl-CoA, one substrate is changing into another succinic acid, and in a coupled reaction this GTP will be converted into ATP. Now this succinic acid, so remember till here, from citric acid cycle to succinic acid, 2 NADH plus H plus, 2 CO2 molecules are released, and 1 CO2 is also released in the link reaction. So understand, pyruvic acid has 3 carbon, and all the 3 carbons have come out in form of CO2. So complete breakdown of pyruvic acid has taken place. So succinic acid will convert into malic acid, which is a 4-carbon compound. So no carbon will be coming out in form of CO2, but there will be production of one FADH2 molecule over here. When malic acid finally converts into oxaloacetic acid, here also one NADH plus H plus is produced. So if you notice, my dear students, in one turn of citric acid cycle, there will be release of 2 CO2 molecules, 1, 2 and 3 NADH plus H plus, 1 FADH2 and 1 GTP or ATP through SLP, substrate level phosphorylation. But remember, this TCA cycle will be running 2 times for 2 acetyl-CoA produced from 2 pyruvic acid during pyruvate oxidation, right? So if you notice, in one turn of TCA cycle, total what will be produced? 2 CO2 molecule, 3 NADH plus H plus, 1 FADH2 and 1 GTP or ATP, you can say. So this is for one turn. You have to multiply it by 2 for the 2 turns. For every glucose molecule there will be 2 turns. So this is your link reaction or pyruvate oxidation and TCA cycle. So let us do some calculations now, right? How many ATP, how many FADH2 and how many GTP or ATP molecules will be produced after or by considering glycolysis, link reaction and the TCA cycle. So let's start with link reaction first. So there are 2 pyruvic acids that will participate, and as a result there will be production of 2 molecules of acetyl-CoA which will conduct or which will enter into 2 cycles of or 2 turns of TCA cycle. There will be production of 2 NADH plus H plus molecules and 2 CO2 at the end of link reaction. Now in the Krebs cycle, what will happen? In every turn there is production of 2 CO2, 3 NADH plus H plus, 1 FADH2 and 1 GTP. So you have to multiply it by them because there is production of 2 acetyl-CoA. So 2 turns of TCA cycle will take place. So total there will be production of 4 CO2 molecules, 6 NADH plus H plus, 2 FADH2 and 2 ATP or GTP. So finally, if you sum up, guys, please notice this. So if you sum up link reaction plus TCA cycle, तो अगर आप लिंक रिएक्शन और TCA साइकिल में बनने वाले प्रोडक्ट्स को सम अप करते हैं, then you will get 8 NADH plus H plus, 2 FADH2 and 2 ATP. This is something you got to remember. Right now, if you sum up the products of glycolysis in this, so in glycolysis there is production of 2 NADH plus H plus and 2 ATP ka net gain. So this there will be, so adding products of glycolysis. So finally, if somebody asks you how many ATP, CO2 and NADH plus H plus and FADH2 molecules are produced at the end of aerobic respiration, including glycolysis, link reaction and TCA cycle, for one molecule of glucose, there will be 10 NADH plus H plus, 2 FADH2 and 4 ATP. ये दोनों ही numbers आपके लिए important हैं. So you have to first understand ये numbers कैसे आए, and then you have to remember also for easily and fastly giving the answers. So remembering them will give you far better speed and accuracy, but you got to understand how these numbers came so that by chance if you forgot, you know how to calculate it and avoid the negatives. So that's the summary. Okay, so now we will talk about terminal oxidation. That means the total 10 NADH plus H plus and 2 FADH2 molecules that we have produced at the end of aerobic respiration. Remember, 10 NADH plus H plus and 2 FADH2. So remember these 10 NADH plus H plus and 2 FADH2 molecules, what are these? These are actually high energy chemical intermediates. Lot of energy is stored in the bonds of NADH plus H plus and FADH2 molecules. These energy stored in their bond is higher than the energy that is stored in 1 ATP. So now finally, during terminal oxidation, what we will do? We will oxidize NADH plus H plus and FADH2 molecules with the help of ETS that is present in the inner mitochondrial membrane, and this oxidation of these NADH plus H plus and FADH2 will lead to production of H plus ions, protons, electrons and energy. So protons, energies will be utilized to produce a strong proton gradient between inner and outer mitochondrial membrane, right? And the electrons will run into the ETS. It will be carried with the help of, from one complex to another, it will be carried. My dear students, there are several electron carriers present within these complexes that is going to carry the electron, and finally it will be given to the oxygen molecule, right? So let us try to understand how this oxidation of NADH plus H plus and FADH2 molecules takes place, right? But before that, let me explain you what are the carriers, electron carriers in the ETS. So electron carriers in ETS, ETS is present in inner mitochondrial membranes. So FMN, FES, ubiquinone and various cytochromes, these are the electron carriers that are part of several complexes present in the ETS running in the inner mitochondrial membrane. So these are the complexes, my dear students. So ETS consists of 4 complexes through which electrons will run, electron will be carried, and there is a 5th complex known as ATP synthase. So ETS consists of 4 complexes that are going to help in transport of electron, and the 5th one is the ATP synthase. See here. So these are the complexes, they all are present in the inner mitochondrial membrane. Complex 1, 2, 3 and 4. All these are going to help in electron transfer. But electron will not be transferred with the help of complex 5, because complex 5 is made up of F0 and F1 particle, and F0 particle will help in facilitated diffusions of the protons from intermembrane space back into the mitochondrial matrix. So they will actually help in the movement of protons and not the electrons. So let us also try to understand what are the components and names of these complexes. Complex number 1 is NADH dehydrogenase, where your NADH plus H plus will get oxidized. Complex number 2 is your succinate dehydrogenase, and at complex number 2 your FADH2 molecule will be oxidized. NADH dehydrogenase has component FMN, FES. Succinate dehydrogenase has FES. Next, complex number 3 is cytochrome B C1, and it has cytochrome B, C1 and FES. Then complex number 4 is the cytochrome C oxidase. Yeah, this is cytochrome C oxidase. Okay, do not, there should be some space between cytochrome C and oxidase. Next, it will have cytochrome A, A3, copper center A and B. So copper centers are present in complex number 4. This is also important for you. Complex number 5th is the ATP synthase name, and it has 2 components, F0 and F1, and F1 will actually have ATP synthase enzyme. This enzyme is going to catalyze the ATP synthesis reaction, and F0 will act as a transmembrane channel through which the protons will move back from intermembrane space to the mitochondrial matrix. So now let us try to understand how ETS works and how oxidation of NADH plus H plus and FADH2 molecules takes place. So listen this carefully. This is your complex number 1, where NADH plus H plus molecules will be oxidized. This is your inner mitochondrial membrane. This is your intermembrane space between outer and inner, where proton gradient will be built. So proton gradient आपका यहाँ बनेगा. Here your proton gradient is getting built. So when NADH plus H plus molecules is getting oxidized at complex number 1, that is NADH dehydrogenase, it will lead to production of protons, electrons and energy. Now these protons will be pumped into the intermembrane space that will help in building up a strong proton gradient. Remember, according to chemiosmotic hypothesis, to produce ATP energy there should be chemiosmosis, and for that there should be a proton gradient. So what are we doing? We are oxidizing NADH plus H plus and FADH2 molecule, जिससे finally क्या होगा, जिससे एक strong proton gradient बन जाएगा हमारा intermembrane space में, जो कि एक बहुत important और prime requirement है ATP बनाने की through chemiosmosis according to Peter Mitchell's chemiosmotic hypothesis. ठीक है। तो अब यहाँ पे इलेक्ट्रॉन मिला आपको, प्रोटॉन और एनर्जी मिली। तो प्रोटॉन और एनर्जी की जो प्रोटॉन्स मिले एनर्जी की हेल्प से उनको पंप कर दिया जाएगा कहाँ पे, इंटरमेम्ब्रेन स्पेस में, और इलेक्ट्रॉन्स को दे दिया जाएगा इलेक्ट्रॉन कैरियर्स को जो कि प्रेजेंट है कॉम्प्लेक्सेस के अंदर। सो फ्रॉम कॉम्प्लेक्स नंबर 1 द इलेक्ट्रॉन विल फ्लो एंड विल बी गिवन टू यूबिक्विनॉन। अब यूबिक्विनॉन जो है वो प्रोटॉन्स को भी उसको रिक्वायरमेंट होती है रिड्यूस होने के लिए along with the electron, so it will take some protons also, right? Now what is going to happen is, my dear students, when ubiquinone, when it accepts electron protons, it get reduced, it converts into a ubiquinol, from quinone to ubiquinol. Now this ubiquinone is going to give, my dear students, the electrons to the third complex number 3, that is cytochrome Bc1. Now notice this, my dear students, the electrons can be passed on from ubiquinol to the cytochrome Bc1, which is the third complex. Protons will not be accepted. So the protons that ubiquinol has, they will be pumped into the intermembrane space and they will help in adding the protons into the proton gradient and building a strong proton gradient. Now the electrons will be accepted by the complex number 3, are traveling into the complex number 3, the electrons will be given to cytochrome C. Now the cytochrome C is a small mobile protein, right, electron carrier, which is going to accept the electron from complex number 3 and give out to complex number 4, right. So finally electrons are reaching to complex number 4. So this is how, my dear students, whenever one NADH plus H plus molecules breaks, right, or get oxidized, the electrons will be traveling from complex number 1 to complex number 3 with the help of ubiquinol, and from complex number 3 to 4 with the help of cytochrome C. And during this whole transport or whole oxidation of the NADH plus H plus, there will be addition of protons into the intermembrane space that will lead to building of the proton gradient, right. So इस तरीके से एक NADH plus H plus मॉलिक्यूल के ब्रेक होने से कई सारे प्रोटॉन्स जो हैं वो इंटरमेम्ब्रेन स्पेस में ऐड हो जाते हैं, जिससे एक स्ट्रॉन्ग प्रोटॉन ग्रेडियंट बनता है। तो इस तरीके से कई सारे NADH plus H plus और FeDH2 मॉलिक्यूल्स ब्रेक होते रहेंगे, राइट, और प्रोटॉन ग्रेडियंट बिल्ड होता रहेगा। प्रोटॉन ग्रेडियंट को बिल्ड करने में, एक स्ट्रॉन्ग प्रोटॉन ग्रेडियंट को बिल्ड करने में वो कॉन्ट्रीब्यूट करते रहेंगे। कौन? FeDH2 और NADH2 मॉलिक्यूल्स, राइट। अब ध्यान से सुनना मेरे प्यारे बच्चों, slowly क्या हो रहा है, because of oxidation of NADH plus H plus and FeDH2 molecules, a strong proton gradient is getting built up. So after some time these protons cannot live in that small tiny space, my dear students, for a very long period of time, and you know that more and more protons are getting added. तो कुछ समय के बाद में ये वहाँ पे जो पूरा सिस्टम है वो अनस्टेबल हो जाता है, यहाँ से और ज़्यादा H प्लस आयंस यहाँ पर स्टोर नहीं किए जा सकते हैं। इसका मतलब अब प्रोटॉन्स को यहाँ से निकालना पड़ेगा। सो एट दिस टाइम द प्रोटॉन ग्रेडियंट विल ब्रेक। सो व्हेन प्रोटॉन ग्रेडियंट विल ब्रेक, आपको पता है प्रोटॉन ग्रेडियंट बनाने के लिए आपको एनर्जी लगानी पड़ी थी, प्रोटॉन्स को पंप करना पड़ा था प्रोटॉन ग्रेडियंट बनाने के लिए इंटरमेम्ब्रेन स्पेस में। तो इसका मतलब जब इस प्रोटॉन ग्रेडियंट को बनाने के लिए आपने एनर्जी दी थी, तो जब अब ये प्रोटॉन ग्रेडियंट ब्रेक होगा तो इसके ब्रेक होने से भी एनर्जी रिलीज होगी, और इसी एनर्जी का यूटिलाइजेशन करके मेरे प्यारे बच्चों एडीपी और इनऑर्गेनिक के बीच में बॉन्ड बना के हम लोग एटीपी मॉलिक्यूल्स बनाएंगे। So this proton gradient will break and the proton molecules will move back. These proton molecules will move back into the matrix. From intermembrane space they will move back into the matrix. Now this membrane, my dear students, the inner mitochondrial membrane is not permeable for charged particles like protons. So these protons cannot come out from any random place. They will come out only from a transmembrane channel. In complex number 5 there is an F0 particle. So in complex number 5 there is an F0 particle that is going to act as a transmembrane channel. So with the help of this transmembrane channel, this integral proteins, the F0 particle, from this F0 particles the protons will do facilitated diffusion. They will diffuse from the intermembrane space back into the matrix. So whenever the protons are going to move back, this is known as chemiosmosis. These protons are moving through the intermembrane space, from intermembrane space into the matrix using the transmembrane channel. So this is chemiosmosis. Now this chemiosmosis of protons is going to release energy, and that energy will be utilized to form bond between ADP and inorganic phosphate to produce ATP. So ATP का production हो जाएगा during chemiosmosis, right? And there is an enzyme known as ATP synthase enzyme which is present in the F1 part. So that complex 5 has F0 and also the complex 5 has F1. So in F0 is going to act as a transmembrane channel and help in facilitated diffusion of the protons. The F1 is going to have the ATP synthase enzyme which is going to catalyze this reaction of ADP inorganic phosphate converting into ATP. Next, so you have seen, my dear students, the role of the oxygen. So oxygen का role यहाँ पर है. Role of the oxygen is towards the terminal, right? So towards the terminal process you can see the role of the oxygen, or oxygen actually participating. But let me tell you, without oxygen, right, in the absence of oxygen, the aerobic respiration cannot take place. This is something very important. So although आप oxygen का role टर्मिनल स्टेज पे देखते हैं, बट ऑक्सीजन की प्रेजेंस में ही आपका एरोबिक रेस्पिरेशन हो सकता है. विदाउट ऑक्सीजन प्रेजेंस पायरुवेट ऑक्सीडेशन, क्रेब्स साइकिल, टीसीए साइकिल कैन नॉट टेक प्लेस. ऑक्सीजन इज द फाइनल हाइड्रोजन एक्सेप्टर. इसीलिए कहा जाता है प्रेजेंस ऑफ ऑक्सीजन वाइटल है इस पूरे रेस्पिरेशन, सेलुलर रेस्पिरेशन या एरोबिक रेस्पिरेशन प्रोसेस में. ऑक्सीजन का जो इंपॉर्टेंस है वो बहुत ही वाइटल है. ऑल्दो उसका यूसेज आप टर्मिनल स्टेज में देखते हो, पर उसका प्रेजेंस बहुत ही ज़्यादा वाइटल है फॉर एरोबिक रेस्पिरेशन टू टेक प्लेस, बिकॉज इट ड्राइव्स द होल प्रोसेस बाय रिमूविंग हाइड्रोजन फ्रॉम द सिस्टम. ऑक्सीजन एक्ट एज अ फाइनल हाइड्रोजन एक्सेप्टर. नेक्स्ट देखते हैं मेरे प्यारे बच्चों केमियोस्मोसिस. ये ऑल्दो मैंने आपको समझा ही दिया, इसको और ज़्यादा अच्छे तरीके से अब और फिर से समझा देते हैं. केमियोस्मोसिस का मतलब होता है क्या? मूवमेंट ऑफ चार्ज पार्टिकल्स. अभी कौन कर रहा है? प्रोटॉन्स. सो मूवमेंट ऑफ प्रोटॉन्स फ्रॉम intermembrane space back into the matrix through the transmembrane channel, and this will be along the electrochemical gradient. ये आप देख सकते हैं, this will be along the electrochemical gradient, right? So ये आपका intermembrane space है जहाँ पे आपका protons है. ये आपका इंटरमेम्ब्रेन स्पेस है जहाँ पे आपके ये प्रोटॉन्स हैं, और ये प्रोटॉन्स विद द हेल्प ऑफ F0 पार्टिकल जो कि ट्रांसमेम्ब्रेन चैनल की तरह काम कर रहा है इन कॉम्प्लेक्स नंबर 5, इट विल एक्ट एज अ ट्रांसमेम्ब्रेन चैनल थ्रू विच फैसिलिटेटेड डिफ्यूजन ऑफ प्रोटॉन्स कैन टेक प्लेस. प्रोटॉन्स कैन मूव फ्रॉम इंटरमेम्ब्रेन स्पेस बैक इनटू द मैट्रिक्स. तो जब ये प्रोटॉन्स मूव करते हैं तो प्रोटॉन ग्रेडियंट ब्रेक होता है, एनर्जी रिलीज होती है. उस वजह से F0 F1, ये आपका F0 है, ये आपका F1 है. तो जब प्रोटॉन्स यहाँ से मूव करते हैं तो आपका प्रोटॉन ग्रेडियंट ब्रेक होता है. प्रोटॉन ग्रेडियंट के ब्रेक होने से क्या होगा मेरे प्यारे बच्चों? प्रोटॉन ग्रेडियंट ब्रेक होने से एनर्जी रिलीज होगी, और उस एनर्जी रिलीज से F1 पार्टिकल की कॉन्फॉर्मेशन में चेंज आएगा. इट विल स्टार्ट रोटेटिंग लाइक अ मोटर. सो बिकॉज ऑफ द कॉन्फॉर्मेशनल चेंज विच इज कमिंग इन F1, there will be, my dear students, production of ATP molecules. ADP और inorganic phosphate के बीच में bond बन जाएगा और ATP molecules का production हो जाएगा, and this reaction will be catalyzed by ATP synthase which is present in the F1 head portion of this fifth complex. So note, my dear students, complete oxidation of NADH plus, one molecule of NADH plus H plus will form 3 ATP, and while one FADH2 molecule when get oxidized, it will produce 2 ATP molecules. Why? Because एक NADH plus H plus और एक FADH2 molecules जब oxidize होते हैं तो they contribute different number of protons into the proton gradient. तो इस वजह से 1 NADH plus H plus corresponds to 3 NADP, 1 NADH plus H plus molecule corresponds to 3 ATP and 1 FADH2 corresponds to 2 ATP. नेक्स्ट बात करते हैं मेरे प्यारे बच्चों रेस्पिरेटरी बैलेंस शीट की. तो बात करते हैं कितने ATP अलग अलग स्टेप्स पर बन रहे हैं. सो ग्लाइकोलिसिस में डायरेक्ट मीन्स सबस्ट्रेट लेवल फॉस्फोराइलेशन से आपको 4 ATP मिलते हैं. याद कीजिए 7th और 10th स्टेप पे. फिर आपको 2 मॉलिक्यूल्स ऑफ NADH plus H plus मिला था 6th स्टेप पे, और हर एक NADH plus H plus इज इक्वल टू 3 ATP. सो टोटल 6 ATP. नेक्स्ट इन द लिंक रिएक्शन में क्या हुआ था? 1 NADH plus H plus in every link reaction, it will take place twice for 1 molecule of glucose because 2 pyruvic acids are produced at the end of glycolysis. So 2 NADH plus H plus 1 is equivalent to 3 to total 6. Next, citric acid cycle or TCA cycle, it will run twice for one glucose molecule. So there will be production of 6 NADH plus H plus. तो 6 को आपको यहाँ पे मल्टीप्लाई करना पड़ेगा 3 से, यानी कि 3 ATP molecules. There will be production of 2 FADH2 molecules, one in each turn of citric acid cycle. So 1 FADH2 equivalent to 2 ATP. So यहाँ पे total 4 ATP will be produced. Next, so 1 FADH2 molecule के ब्रेक होने से, ये H ज़्यादा आ गया, एक FADH2 मॉलिक्यूल के ब्रेक होने से आपको 2 ATP मिलता है. यहाँ पे 2 है तो 2 into 2 it will be 4. एंड डायरेक्ट गेन SLP होता है 2 ATP का GTP बनते हैं. इन कपल्ड रिएक्शन इट कन्वर्ट्स इनटू एन ATP. सो अगर आप इसको सम अप करें, so it will lead to 40 ATP, right? So total for one glucose molecule there is total production of 40 ATP at the end of terminal oxidation or at the end of terminal stage, right? So you include glycolysis, link reaction, TCA cycle, सब कुछ खत्म करने के बाद एक ग्लूकोज है, टर्मिनल ऑक्सीडेशन करने के बाद में, यानी NADH plus H plus और FADH2 मॉलिक्यूल्स को ऑक्सीडाइज करने के बाद में आपको टोटल गेन 40 ATP का होता है. लेकिन याद कीजिए ग्लाइकोलिसिस के अंदर ऑन स्टेप नंबर 1 एंड ऑन स्टेप नंबर 3 2 ATP का कंजम्पशन भी हुआ था. तो कंजम्पशन होने की वजह से यू हैव टू माइनस 2 ATP फ्रॉम हियर. सो टोटल 38, यानी कि टोटल 38 ATP मॉलिक्यूल्स आर प्रोड्यूस्ड और कैन बी प्रोड्यूस्ड फ्रॉम वन मॉलिक्यूल ऑफ ग्लूकोज. नेट गेन इतना हो सकता है. नेक्स्ट बात करते हैं मेरे प्यारे बच्चों एम्फीबॉलिक पाथवे. तो ध्यान से सुनिएगा. सबसे पहले रेस्पिरेटरी सबस्ट्रेट ऐसा नहीं है कि सिर्फ कार्बोहाइड्रेट या ग्लूकोज ही हो सकते हैं, प्रोटीन्स और फैट्स भी हो सकते हैं. बट व्हेनेवर प्रोटीन्स एंड फैट्स आर acting as a respiratory substrate, they might not follow the exact pathway which the glucose has followed. So ज़रूरी नहीं है कि proteins and fatty acids will start from glycolysis. They might start somewhere in the middle of the glycolysis or from link reaction or directly from TCA cycle or somewhere in the middle of the TCA cycle also. So let's check out. So whenever your fats are acting as a substrate, respiratory substrate, they will break down into fatty acid and glycerol. Glycerol will finally change into phosphoglyceraldehyde, right? PGAL. So आपका DHAP जो है, आपका slip of tongue. What I wanted to say over here is आपका जो glycerol है, I repeat myself, आपका ये जो glycerol molecule है मेरे प्यारे बच्चों, glycerol molecule will finally convert into PGAL and then it will continue the glycolysis, will do the link reaction and Krebs cycle. तो यानी glycerol आपका समवेयर इन द मिडिल ऑफ ग्लाइकोलिसिस से एंटर करेगा. फिर बात करते हैं फैटी एसिड. फैटी एसिड विल कन्वर्ट इनटू एसिटाइल कोए एंड इट विल डू द क्रेब्स साइकिल. फैटी एसिड ग्लाइकोलिसिस और लिंक रिएक्शन को स्किप कर देगा. प्रोटीन्स सबसे पहले अमाइनो एसिड्स में कन्वर्ट होंगे, फिर उनमें डीएमिनेशन होगा, अमाइनो ग्रुप को रिमूव करेंगे. अब डिपेंडिंग ऑन द स्ट्रक्चर ऑफ द अमाइनो एसिड, तो या तो ये अमाइनो एसिड कैन फॉर्म अ पायरुविक एसिड and will undergo link reaction and enter the TCA cycle, or from this amino acid directly acetyl CoA can be synthesized and only Krebs cycle will take place, or this amino acid depending on its structure can also start or can also enter the respiratory pathway somewhere in the middle of the Krebs cycle. So ये depend करता है उस amino acid के structure पर. तो इस तरीके से दूसरे respiratory substrate enter करते हैं respiratory pathway के अंदर. अब ध्यान ये देना है कि रेस्पिरेटरी पाथवे को आप एम्फीबॉलिक क्यों कहते हैं. एम्फीबॉलिक का मतलब एनाबॉलिक भी है और ये कैटाबॉलिक भी है. तो हमें ये तो पता ही है कि रेस्पिरेशन इज अ कैटाबॉलिक प्रोसेस बिकॉज रेस्पिरेटरी सबस्ट्रेट्स आर ब्रेकिंग ओवर हियर. बट हाउ respiration can also be an example of anabolism. So listen this carefully my dear students. Right now you can see how these respiratory substrate, fats, glycerol, fatty acid, carbohydrates, proteins are breaking. But at the same time, if in the cell during metabolic reactions, in any of the metabolic pathway, if a cell needs to produce protein molecules, so in that my dear students, during formation of the protein molecules from the respiratory pathway, either pyruvic acid my dear students or acetyl CoA, they can be withdrawn, right? So pyruvic acid or acetyl CoA can be withdrawn from the respiratory pathway in order to produce amino acid, in order to produce proteins. तो हुआ ना ये anabolism का example. उसी तरीके से मेरे प्यारे बच्चों, अगर आपके सेल के अंदर फैट्स बनाने की ज़रूरत है तो रेस्पिरेटरी पाथवे से PGAL or acetyl-CoA will be withdrawn and they will be utilized to produce fatty acid, cholesterol and finally fats. So इस तरीके से during formation of the fats, proteins, carbohydrate, be respiratory pathway come into play and during their breakdown also. So that is why finally we will consider respiratory pathway as amphibolic. That means it includes anabolism as well as catabolism. So finally the last topic we will discuss in this class is respiratory quotient. So remember my dear students, this respiratory quotient is actually a ratio of volume of CO2 evolved divided by volume of O2 consumed during cellular respiration. So RQ equals to volume of CO2 evolved and volume of O2 consumed during cellular respiration. Now you have to consider that the respiratory substrate has oxidized completely. So after complete oxidation of a particular respiratory substrate, its RQ value is going to vary. So listen this carefully. Whenever your respiratory substrate is a carbohydrate, let's say glucose, the RQ is going to be 1. So whenever your respiratory substrate, guys, whenever your respiratory substrate is carbohydrate, example glucose, you can see the volume of CO2 evolved will be equal to the volume of O2 consumed. So 6 CO2 will be evolved and 6 O2 molecules will be consumed. So RQ value is 1 for carbohydrates. Remember. Now if you change the respiratory substrate, like for fats and proteins, the RQ will be less than 1. So fats, if I talk about my dear students, like tripalmitin, triolene, if I talk about, the RQ will be somewhat around 0.7. So RQ for the fats is 0.7 and whenever the proteins are acting as a respiratory substrate, then the RQ is going to be 0.9. तो RQ आपका फैट्स के लिए है 0.7 और RQ आपका प्रोटीन्स के लिए है 0.9. So whenever you change the respiratory substrate, RQ will also change. For fats 0.7, for proteins 0.9 and for carbohydrate it will be 1. So that's all from this class my dear students. Thank you for listening. In this lecture we have completed the entire respiration in plants. If you like this lecture, give me a thumbs up and also let me know in the comments how did you find this lecture. Till now, if you haven't subscribed to Infinity Learn, my dear students, do considering subscribing to Infinity Learn NEET channel so that in future whenever we will go live or upload videos like this, you will also be notified. So for that, also do not forget to hit the bell icon. Like, share and subscribe with all those students who are preparing for the NEET 2023 exam. All the best.